let's move on now to Rebecca. Rebecca Ram is an independent scientific research consultant with an MSc in toxicology and a BSc in applied biology. After a decade working in clinical trials in the pharmaceutical industry, she changed paths to focus on alternatives to animal testing and the promotion of human relevant research, as well as some continued clinical projects, for example, the 100,000 Genomes Project. Rebecca has worked or provided scientific support to a number of scientific and animal protection organizations, most recently the Lush Prize and the Safer Medicines Trust. And I think Rebecca's going to talk some more about the absence of a level playing field, which Katie talked about, but also about how the cosmetics bans have provided an incentive, a push for the development of alternatives. So over to you, Rebecca.
Thanks, David. I'll just share my screen.
While you're doing that, Rebecca, I'm pleased to see that Jared has now joined us. Look forward to your talk, Jared.
Thank you. Yes, glad to be here.
Sorry, everyone. I'm just having a moment trying to share the screen. Bear with me. Okay. There we go. Can you see my slides?
Yep.
Great. Thanks. Okay. Thanks, David and Dee Dee, and the opportunity to present tonight for this webinar. So as you can see there, my talk will focus on ending animal testing and advancing human relevant research via what are called new approach methodologies, which I will explain, and the funding and regulatory solutions. And I'll draw on some points that David and Katie have already made and recap some of those, but provide some new perspectives as well. So just to recap something that David spoke of in my introduction, I represent two organisations this evening. One is the Lush Prize, which is an award scheme for initiatives which work towards the replacement of animals in testing and biomedical research. And I also represent this group, the Safer Medicines Trust, which is a patient safety organisation. And again, one of their missions is to promote human relevant research in the form of animal-free methods. And Safer Medicines Trust is also a founding member of this group, the Alliance for Human Relevant Science. So as well as working in clinical trials, I've also had the privilege of working with all of these organisations in the past too. So it's been an invaluable experience. So just briefly, what I'll cover, an introduction and just a word on some terminology. And then to go on, as David said, to talk about some of the differing standards in animal methods versus alternatives. I'll cover aspects of funding and regulatory acceptance and harmonisation of regulations and guidance. And again, leading into what we perceive as a lack of a level playing field. And then go on to talking about a legal framework and relevance to the audience this evening, and deadlines for the implementation of NAMs. Again, I'll explain that. And then the EU cosmetic testing bans as a driver of change, and perhaps as a working model, and finishing with some positive change and conclusions. So as you've already heard from David and from Katie, the 3 R's broadly are reduction, refinement and replacement. So they are, as we know, called alternatives, which can in some respects be confusing or misleading because, as you've already heard, 2 out of 3 of those, reduction and refinement, still use animals. So there's been a push in recent years for some organisations to use terms which are clearly more focused on replacement. So the 1R, for example, is a term that the Lush Prize has used quite frequently to show that they are invested in replacement-only technologies. And again, the term alternatives, it is very broad, and we do need to use it because it is recognised in a lot of legislation, so we have to be consistent. But a personal opinion of mine is that we could perhaps be using the term more like solutions moving forward where it's appropriate, because the new methods that we have are providing solutions where certainly some animal models haven't in the past. So from here on in, for the rest of my presentation, I'll use this term, which is New Approach Methodologies, or NAMs, to represent animal-free methods or alternatives. So when we talk about New Approach Methodologies, what do we mean? I won't go into too much detail on these because it may be a bit overwhelming if they're new topics, but these three categories represent broad areas where NAMs have really come along in advance over the last decade or more. So adverse outcome pathways. An AOP is basically the mapping of a chemical and the effects it has from a very basic molecular level through cellular and organ effects and through to the adverse events it can have in an individual or even a population. Then we have what are called microphysiological systems or organ on a chip. These have been developed over recent years for different organs, for example, lung on a chip, liver on a chip. And as you can see here, this is a tiny, very simple looking device. And it has been built with microchannels in it, which are perfused with human tissues and cells. And they're used to apply chemicals to mimic the effects that humans would have. And then we have here what are called computational approaches, or in silico, as they're often termed, and you may know that term. That includes computer-based models themselves of analysis, but also this term big data, which is basically the concept of making use of the vast amounts of data that are already there. And that's vastly underused at the moment. So going on to a comparison slide here, I hope this doesn't look too busy. I have tried to limit the text, but to compare the differing standards and attitudes applied to animal methods versus NAMs. So I've got down the side here, and I know Katie covered some of this as well, very well. So we've got here validation, performance, funding, and regulatory acceptance. Now, animal methods have had years of convention. They've very much been grandfathered through in some respects, and they're considered traditional. And by many, mainly those that use them, they're still considered a gold standard. Whereas NAMs have faced a lot more resistance, with a large proportion of the argument being that they're not whole animals. Animal methods have never been formally validated, and unfortunately there is institutional bias which exists, and applications to use animals are very rarely, if ever, rejected. We had a good question on that early on in the chat, and we'll come on to discuss that more, but they really are. It's rather a self-driven system with animal project licensing. As long as personal and establishment licenses are in place, this is a very demand-driven system. Whereas again, for NAMs, it's a long and challenging peer review and validation process. It can take years, and there is rejection in there, with a major obstacle being that those that develop new methods are asked to go back and perform them in animals. Regarding performance, animal methods are well known to have limited ability to predict human response, and there's this term of lack of bench to bedside translation, and they have many unmet needs in major diseases, many listed there, and far more. Whereas combined NAMs provide a more human relevant level of data for particular endpoints on a critically more basic level, more molecular level. And again, Katie and David alluded to this, where an argument is that there's a lack of like-for-like replacement, where that would never be the case because NAMs are much more targeted, and many NAMs would replace one animal test, which is better for human disease research. Again, better science is what we're aiming for here, rather than alternatives or replacements of flawed methods in the wrong species. Funding sources can often be routine and repetitive. We see that in project licenses and in the summaries of animal research projects. And equally difficult is the gaining of NAMs funding on the flip side of that. So this leads to, in terms of regulatory acceptance, a high regulatory, and I'll put expectance here, of animal data, because I thought it was sometimes a more appropriate term. Animal data is expected. and a low regulatory acceptance of NAM data. So this next slide is really just a few quotes and testimonies relating to some of the points on the last slide, again highlighting the differences. So again, that the belief is, and a belief being the key word, that the use of animals is the gold standard, where we've seen bias in the peer review process, perhaps understandably in a way, because many that review and new applications for animal-free methods are animal researchers. So they then request that they need to see the validation, so to speak, in an animal model, which is rather counterproductive. And again, some other quotes here on difficulties to get research funding, and again, a block when working on animal-free methods for decades in some cases, and not getting anywhere with regulatory acceptance. So there is a bit of a deadlock here. Leading on from the first slide, again, just what I have put together here is a diagram really that can be seen as an animal research cycle. It's the chain of repeated use and repeated application and funding of animal research projects, which leads to regular global supply and demand for breeding of animals, security of repeat funding sources, and we see this in animal project licenses all the time, where, for example, this animal work will lead to future funding for further experiments. So we know there is a real sort of perpetual system going on here. These quotes down the side again are from testimonies and non-technical summaries, as they're called, of animal licenses. Well-established models, well-published models basically means the same animal tests have been done over and over again. There's a lot of data on them, so the researchers will choose to do those tests again. And of course, we know that regulatory protocols often specify particular types of animals. So summarising here, three main sectors, and Katie talked about these as well: cosmetics, industrial chemicals, and pharmaceuticals. A big problem at the moment is within cosmetics on the left there, the conflict between cosmetics regulation, which enforces a ban on animal tests, versus other regulations such as REACH, which still require animal tests. As a result, the cosmetic regulation is being undermined, and products which, of course, many of the public would consider to be cruelty-free have actually contained animal tested ingredients. Industrial chemicals, again, with REACH among others, as it's a major piece of chemicals legislation, animal tests are widely considered inefficient to keep up pace with the high volume of assessment that's required of so many substances. And there's still a big unknown on the total number of chemicals worldwide, and concerns over insufficient data packages, despite all of these regulations. And then with pharmaceuticals, there is a very high failure rate within clinical trials. I can speak from experience of that with working in pharma for several years. It was quite an eye-opener. And between 86 and 90, and sometimes higher, 90% of new drugs fail clinical trials somewhere during the clinical trial phase, having been successful in preclinical tests, many of which include animal studies. Liver toxicity is a major cause for post-market drug withdrawal, and 1 in 3 of those are due to liver tox. Many diseases still lack effective treatments, and as I've said, the pharma pipeline, the attrition or the rate of drug approval is in crisis, with costs going up and new medicines not keeping up with those increased costs. And when we look at animal tests and how they're used and the failure rates, it does beg the question how many potential treatments have been overlooked. So moving on, with a lack of harmonisation again, and lack of level playing field generally, and recapping some of what Katie covered earlier, some examples here, a few more examples. The abnormal toxicity test, the ATT, it's pretty much considered useless now. It's been deleted in many areas, as I've listed there, but still requires agreement from regulators worldwide to make a concrete deletion of this test. Another prominent example is a particularly GRIM test, a 1-year chronic toxicity test for pesticides in dogs. It's been banned in the EU and the US, and there's been a 10-year campaign to ban it in other countries. But Japan still requires it. So companies still use the test to meet worst case requirements, which reinforces the urgent need that NAMs must be globally accepted by all regulators. Otherwise, companies, perhaps understandably in some ways, they need to see what the worst case is that they need to meet, so they'll still do the animal test. And it's a dreadful case that's slipping through the cracks. Conflict between regulations, as we've talked about, cosmetics and the REACH regulation, which has led to an excellent campaign for one substance, one registration. And at the bottom of this slide, I've referenced a really useful webinar that was held recently on that by some of the lead campaigners on it. And again, recapping some of Katie's points, it's an incredibly slow process. So even when agreement is reached to use NAMs, updating the regulations can take years and lack proper regulatory enforcement. So again, some of the OECD approved skin sensitization methods were developed, but animals are still used in skin sensitization tests, and Katie gave some really good figures on that. And human skin models validated and approved, and yet the animal tests weren't removed from guidelines until a decade later. So there's a whole heap of issues here to improve, to make sure that animal tests are replaced. I put this slide in because there are a couple of surprising points in that there are very few legal requirements for animal tests, starting with the vast majority of animal procedures aren't legally required, and they're not retrospectively assessed. So briefly, within the EU Animal Experiments Directive, there is provision for member states to apply retrospective assessment to animal experiments that have been carried out. But sadly, the take-up rate for that and the actual number of tests that are retrospectively assessed is shockingly low. Over 80% aren't even assessed to see if they met their scientific objectives or to look at the animal welfare. And that's a rough guide for the UK and EU. Another important point, no animal test is legally mandated for medicines. There's no categorical requirement to use animals. And also there's no second species mandate, which is sometimes quite surprising for people to hear. It's all due to a guidance framework and the regulatory guidelines for medicines, which I'll come on to talk about briefly. Again, REACH is quite a prominent example. It promotes the use of NAMs, but also requires animal tests where no alternative is available. So there's conflict within the regulation as well as with other regulations. And it's important to understand from the point of view of regulators, the problem with risk aversion. There's a lot of nervousness. It's understandable. There's a lot riding on clinical approval of drugs, and no one wants to be the first. And we hear that time and time again. No one wants to sort of be the first to stick their head above the parapet, so to speak. So due to a lot of those, a combination of these factors, regulatory and academic convention continues and relies on animal testing despite major limitations. So I put this slide in because we do hear this in terms of attitudes from regulators in discussions do vary, it should be said, but there are those that really do adhere to the regulations and don't really feel they should ever be questioned, even when they're in a position to do so themselves. So I thought it was appropriate to put that in. Discussion with government and regulatory agencies. Again, we're at a bit of it might be fair to say we're at a bit of an impasse on some of it, because just taking this blue box at the top for now, lack of funding of NAMs is a big problem. It's even more concerning that in chats with some funding agencies and regulatory bodies, it's not really considered an issue. And instead, the issue is considered to be that the methods aren't good enough, that the NAMs aren't up to the job. And of course, to a certain extent, that's true, because they're not all there yet. More work needs to be done. They've not had the funding or the attention that they should have, and the lack of level playing field, as mentioned earlier. But the fact that those methods aren't good enough yet, or certainly some of them, comes back to needing funding, because the first thing that's needed to develop and optimize a method is greater funding. It's also linked to greater transparency on gaps that require filling and what's missing. And this comes back to the regulators, and to start with them and ask the right questions on what's needed and from the right angle. Again, certain regulators will say that they have an open door policy on discussion about this, and that needs maximising and using to the fullest, because some regulators have even said that over adherence to animal test guidelines is not what they're looking for either, yet animal testing continues. So there's a whole raft of discussions that need to continue here on funding and regulation. Going on again to another point about the disadvantage of animal models, again in the context of discussions with governments and regulators, they eventually reach a dead end, even with many attempts to genetically, surgically or chemically modify animals, and it's an industry in itself now, the use of GM mice, as Katie mentioned. Despite that, it's still competing with the underlying species biology and reaches a dead end eventually. Whereas the advantage of NAMs is that they can continually be improved and optimized, and that presents business opportunities as well as scientific ones for training and empowerment and outsourcing. And to put a positive point within all this negativity, NAMs are being used. Pharma is using them, but it's still rather on a low scale. They are used at the moment more as complements to animal models rather than to replace them. And again, that presents, if there was upscaling from government backing, it would present infinite business opportunities to achieve what's ultimately needed, which is human relevance. And so moving on to the relevance for many listening this evening, a legal framework and deadlines for the implementation of NAMs. And I've put in here the well-known cosmetic testing bans that were phased in between 2009 and 2013, and the argument that they could be used despite the problems we see with them as a legal working model. So a bit of history. The campaign originated back in the 70s, starting with public support and then growing into effective lobbying. And the political campaign took 20 years, from 1993 till 2013, to achieve the phasing of the bans. Many challenges and delays from companies, interestingly, who tried to stop the bans at the time and are now shouting about them and have got on board, so it's quite interesting. There's overwhelming public support now for the bans to be upheld, stronger than ever. And despite the conflicts with other legislation, they are still a key legal landmark towards ending animal tests. And there are problems at the moment which have caused real frustration for those that have worked on this for a long time. But seeing the positive side, we must remember that the fact that the bans are there are a good thing in themselves. And if they weren't there, things would be a lot worse. So the bans had a domino effect in many other countries, which is fantastic. And many new methods were developed in preparation for the forthcoming deadlines. And despite some still being under development at the time, the final 2013 deadline was still upheld. So it was a real legal landmark. And so using that as a model, perhaps establishing a legal framework to drive all of the points that I have here. Diversion of funds from animal models to NAMs. And I use the word diversion carefully because, of course, talking about increasing in funds shuts a lot of doors, because the first reaction is there's no money available. You know, we've got to find it from somewhere. So let's be more pragmatic and suggest that diversion of funds is more appropriate. The government action on this would achieve benefits for everyone, including the government, but it needs whole-scale action. So business opportunities, more jobs, agency initiatives and training. Timing it right would again mean that early career researchers who are a key audience in this would get on the NAMs roadmap earlier. And sorry, I missed a point there on the regulatory resistance and a lack of confidence, again being risk averse, could be avoided by using pilot studies and trial projects. Again, this is happening, but it's rather on a bottom-up basis rather than a top-down. So we'd need to see much more whole-scale action on that. And then as I've said, to get NAMs in the hands, particularly of young researchers, from the basic and what's called blue sky research stage through to increasing confidence and wider acceptance by the time the regulatory testing phase is reached for new drugs, for example. And so increasing the new generation of knowledge base in NAMs will achieve the scientific transition. But importantly, I think it really needs a legal framework to do that. One more point on the cost savings: the deployment of NAMs would ultimately save money, replacing animal tests by a cost saving of up to 90% per test, which was some analysis done recently by some experts in the field. A question we also see, which is very valid, is that some NAMs, of course, would be more expensive at first with setup and deployment, but we know that the evidence suggests that by the time NAMs are more in stream, they would actually save money and be a lot cheaper, and of course more reliable than animal tests. This figure on the right is a set of measures that were promoted in a very good white paper recently by the Alliance for Human Relevant Science, and I recommend it. It's a very good read. So moving on, I'm coming to my last few slides to keep us to time, hopefully. Some positive change. Again, Katie spoke about some of these as well. The US EPA, the Environmental Protection Agency, has made an announcement recently to eliminate all mammalian studies by 2035. It sounds a way off, but any timeline or timeline in the future that a big agency is willing to make is good news, because they can be held accountable. The US FDA has recently agreed and is now testing organ-on-a-chip technologies, which was quite groundbreaking at the time. From a Lush Prize perspective, we see increasing demand year on year from early career scientists who want to use NAMs, particularly for funding, and we've awarded funding in areas where gaining support is difficult or refused, and for greater recognition within political lobbying, research, training and education. I should mention very briefly the excellent roadmap initiatives that some of us may be aware of that are out there. And the bad news, as Katie again mentioned, is that the number of animals generally hasn't changed. But the good news is that the number of researchers is increasing, and some of that is down to the fact that they're going straight into NAMs. Young researchers don't have the gold standard argument to live up to, or the comparison to animal data that perhaps some more old school researchers have. We see an exponential growth of in vitro and in silico markets, and there is some cross-sector use of some NAMs. Just a point on the two pictures briefly. This one at the top is 3 fantastic researchers from a few years ago that were really struggling to get funding from Japan, Korea and China. The lady on the left, in particular from Japan, had real she had some real challenges in gaining recognition for her work, and the award that she got really helped her get on another platform to continue her research. So it was fantastic to hear that. And just briefly on the bottom here, NAMs at various conferences, any session on alternatives is always really oversubscribed, and there's usually people spilling out the door. So I thought I'd put that in, because people do want NAMs, they are being used, things are happening, but we just need more of a whole scale change. I'm nearly there.
If you could wind up.
Yeah, will do. On my last
minutes, thank you.
Will do, thank you. Yeah, so just to summarise some recent changes in the news, positive changes is being seen politically and in the industry, with it being called a tipping point, because animals are widely considered unnecessary now for next generation safety and efficacy assessment. And this is on the right is a recent press release on paradigm shifts from Unilever and other industry members. And many will know of the recent very promising news on the EU vote to put together an animal replacement strategy. So coming to my very last slide, some conclusions and take-home messages. Again, I'm using the term because it works really well. Encouraging progress is underway, but still a drop in the ocean when we look at what's still going on globally for animal research. Whole-scale government action is needed, driven by political and industry lobbying. Of course, scientific innovation, which to some extent is there, but needs more to continue. Meaningful future deadlines and legal mandates for NAMs will clinch some of this. again, I spoke about the business opportunities that are provided, and this really needs pushing for the business advantages, and call to action to divert funding to see some of those advantages. Removing comfort zones and research silos, they do exist. Changing hearts and minds, all of this is necessary. And as I've said, the law needs to catch up with science. So a solution, again, largely in my opinion, is a robust legal framework to guide all of this. And this diagram I've put on the right-hand side is really just to show about turning around the thinking. So instead of saying we can't use NAMs because of this, we should use NAMs because they provide information that we can't get from animals. And so I'll stop there. I hope I haven't overrun, and say thank you for listening. And of course, I'll take questions in the chat and later, but please email me afterwards if you'd like to. Thanks, David.